Hello students. So today we are going to learn on subtopic 13.2 Biotechnology, a continuation from our previous lesson from the same chapter Genetic Technology. So without further ado, let's dive into it. Let's look at the learning standards. Firstly, you must be able to explain the meaning of biotechnology. Secondly, to describe application of biotechnology in life. There are a few applications that we will cover in this subtopic. And lastly, to justify the importance of biotechnology in life. Now, biotechnology is the use of living organism or substances from living organism to develop or make products in industry. This genetic technology involves the application of living organism to develop agricultural, medical, or environmental products or processes. Now, genetic engineering is an integral part of biotechnology, and most biotechnology processes are classified as fermentation as it occurs in a fermenter, or we call it as bioreactor. Now, biotechnology can be classified based on economic industry which it is applied. Now, the classification uses 10 colors, okay, that each one corresponds to a certain branch of economic industries. So let's start with the first color, red. Red biotechnology represents the medicine and also human health. The blue technology represents marine biotechnology. The gray biotechnology represents the environmental or ecological biotechnology, for example, to maintain biodiversity, to remove pollutants or contaminants using microorganisms and plants. The purple biotechnology represents the patents, publication, invention, intellectual property rights. We're talking about legal, ethical, and philosophical issues that are associated with biotechnology industries. Then we have the yellow biotechnology, which represents the food biotechnology, nutrition science, for example, food production in fermentation, to make wine, to make cheese, and also beer. The black biotechnology represents bioterrorism, okay, biotechnological warfare, biocriminology, and also anti-crop warfare. Then we have the gold biotechnology, which represents the bioinformatics and also computer science. Now, the brown biotechnology represents the biotechnology of desert and dry regions. And green biotechnology involves the improving agricultural products. And lastly, the white biotechnology represents industrial biotechnology. For example, the use of microorganisms in chemical production, designs, and also production of new plastic and development of new energy sources. So, in modern genetic engineering, these are all the application and the industry involved using biotechnology. Okay, let's look at the application of biotechnology in life. So we have here a few that we are going to look into, which is the first one, gene therapy, DNA profiling, production of insect-tolerant plants, cleansing of oil spills. The others we have is production of microbial metabolites, nanobiotechnology, and also bioinformatics. Let's look at gene therapy. Gene therapy refers to a collection of methods that alter or replace a defective gene of a hereditary disease in a child or embryo. It involves the transfer of normal genes into the genetic materials of a cell to replace the defective genes that cause a genetic disorder. As a result, the body is able to produce the right protein or enzyme it needs, and thus eliminates the cause of this disorder. So gene therapy is usually used to cure cystic fibrosis, okay, muscular dystrophy, a few types of cancer, okay, that is related to genetic. So usually a cell with an abnormal gene, okay, is inserted with a normal gene using a virus, and then the cell will be able to function normally. Next, we have DNA profiling. Now, DNA profiling is a technique in which an individual is analyzed to reveal the patterns of certain nucleotide segments in the DNA, which we call as repeats, that do not code for any proteins. So these patterns, okay, are in the form of a series bands called DNA profile, or we call it as DNA fingerprint, and are different for each individual except identical twins. Now, DNA profiling is used to resolve paternity or maternity issues. They are also used in forensic science to identify criminals. Okay, they are also used to identify genetic disorder that may cause hereditary diseases, cancer, and other genetic diseases. And it is also used to determine suitability of organ donor and recipient. What are the steps in DNA profiling? Well, first, the blood, semen, or skin sample is obtained from an investigation scene, for example, a crime scene. It is applicable to any type of investigation that needs to be carried out. So here the DNA is extracted from the sample. Restriction enzyme is used to cut the DNA into different sizes of DNA fragments. Then these DNA fragments of different sizes are then separated by gel electrophoresis. DNA fragments of different sizes are transferred from the gel to a nylon membrane. Then the radioactive probes are added to the nylon membrane. Okay, for example, X-ray film is then placed on top of the nylon membrane. So this X-ray film is processed to show the position of DNA bands that forms the DNA profile. So if you look at this part here over here, based on the DNA profile below, can you determine who is the biological father of the child? So which one it is? So we can actually determine this by looking at the DNA fragments of the mother and the child. Now if you look at the mother has certain bandwidth that is similar to the child. And since the child is a combination of the mother's DNA and also the father's DNA, so we can actually determine. So you look at the purple one is the mother, so it's the same one. Now you look at the red one, we are going to determine where is it same width. So looking at this, looking at the red color, so the same bandwidth here is the father number 1, because there are band that is not in father 2. So the possibility of the father of this child is father 1. Another question here, we have mother and we have child here, but now there is no color to differentiate. But how are you going to determine? So looking at the child DNA is a combination of the mother and the father's fragments. So let's have a look. So mother here, no, child over here. So the same line goes here. So dat2 and dat3 has the same line here. Now the third line, same with the mother. Okay, so forget about it. Then we have this one also same with the mother. What about this 2 here? So the mother doesn't have this band, so it's coming from the father. So here is this one, father 1 and dad number 3. This one is from dad number 3. So the possibility of the father is dad 3 because the DNA fragments which is not similar to the mother is similar to dad number 3. So we always have to see what DNA fragments bands that are shared, okay, from the mother and the father to the child. So let's look at the production of insect-tolerant plants. Now, crop plants such as cotton, brinjal, and maize are prone to attacks by insect larvae, which destroys the shoot and fruits. So biotechnology is used to overcome this problem and to save costs on using pesticide. A gene that expresses insecticidal protein is isolated from a soil bacterium known as Bacillus thuringiensis, or we call it as BT. Okay, BT is for the bacteria Bacillus thuringiensis. So this gene is inserted into the DNA of the brinjal plant or the other plants, any crop plants, and these crop tissues will then express the gene to produce Bt toxin that can kill the insect larva. So when the shoot or fruit of the genetically engineered crop, brinjal, maize or cotton, is ingested by the insect larva, the Bt toxin is released in the insect's body, thus killing the insect. The development of the Bt brinjal or the Bt crop variant has significantly benefited farmers and consumers as well as protected the environment. Okay, let's look at cleaning of oil spills, or we call it as bioremediation. Now we know that oil spill in ocean and land have severely affected the environment, causing both ecological and also economic damage. So bioremediation is a biotechnology method that is used to clean up oil spills in water and also soil. So in this method, bacteria are used to break down oil into less harmful substances such as carbon dioxide and also water. So specifically, a genetically modified superbug, or we call it as Alcarnivorex borcumensis, or also we can use Pseudomonas putida, okay, which was created to enhance the cleaning up of oil spills as it can break down most types of hydrocarbon. So this type of bacterium was created by inserting genes from 4 different types of bacteria that can break down different types of hydrocarbon. So we have here Pseudomonas putida, or we can also use Alcarnivorex borcumensis that is used to treat oil contamination. Other applications of biotechnology. A microbial metabolite is an intermediate or end product of metabolism in microorganisms such as bacteria and fungi. The main microbial metabolites are alcohol or ethanol to be specific, and also antibiotics. Biotechnology is used in the production of alcoholic drinks such as beer and wine, and also in the bread formation through fermentation of yeast. During alcohol fermentation in yeast, ethanol and carbon dioxide are produced. The metabolite ethanol can be used to make alcoholic drinks or to produce commercially for biofuel. In bread making, carbon dioxide traps the dough, makes the dough rise, and makes the bread light and soft. When we talk about antibiotics, antibiotics such as penicillin from the fungus Penicillium and also streptomycin from the bacterium Streptomyces can be produced in the industry by culturing the fungus or the bacterium in a fermenter. So it can be produced in a very large scale. Now, let's look at nanobiotechnology. Nanobiotechnology is the combination or fusion of biotechnology and also nanotechnology. Nanotechnology is a new scientific approach that uses substances at atomic or molecular levels, and nanobiotechnology is mainly applied in medical and also clinical fields. This technology allows the detection of pathogens and diseases at an early stage so that effective treatment can be carried out to prevent the disease from progressing. Nanobiotechnology is also used in drug delivery, drug discovery, and pharmaceutical development. Now, bioinformatics in general is the science of collecting and analyzing complex biological data such as genetic codes. Bioinformatics has many applications in the field of biotechnology. Some of the important applications includes automatic genome sequencing, automatic identification of genes, identification of functions of genes, predicting the 3D structure, modeling and pairing of genes. The importance of biotechnology in life. Biotechnology is important to mankind because of its extensive use in genetic engineering, medicine, agriculture, environment, and various industrial applications. As a result, it has contributed to the progressive growth and development of the production of food, agriculture products, livestock, and medicine, which in turn raised the economic status of the country. So the use of biotechnology also enhances the quality of life for plants, animals, and humans. So some of the importance of biotechnology is in terms of agriculture, food processing, medical, and also environment. In the field of agriculture, biotechnology is used to produce crops that are insect or pest resistant and also herbicide tolerance. This increases the quality and quantity of crop production to sustain the rapid growth of population. So biotechnology also reduces the cost of farming and health risk of consumers as well as farmers. It produces crops that can adapt to climate change, produces crops that contains more nutrients, for example, the golden rice, and many other agriculture uh, advantages. In the medical field, biotechnology produces pharmaceutical products and medicines such as antibiotics, vaccine, and various drugs. It produces large quantities of nutritional supplements such as vitamins and insulins for diabetic patient treatment. And it also used in gene therapy to treat various genetic diseases. In the field of food processing, biotechnology is used to make wine, beer, and bread through the fermentation process of yeast. It produces enzymes from fungi and bacteria which are used in the production of biological washing powder, clear fruit juice, lactose-free milk, and also yogurt. And in the field of environment, biotechnology is used to treat and recycle waste products in order to preserve natural resources and to reduce pollution. For example, the bioremediation bacteria which breaks down oil in water and also soil. It is also used to produce bioplastics which can decompose compared to normal plastic. So, in bioplastic, bacteria is used to produce bioplastic products such as credit cards and also medical adhesive. So this biodegradable plastic actually reduces environmental pollution. Now, let's check our understanding of what we have learned from the lesson. The first question, the use of living organism to break down environmental pollutant is called microremediation, phytoremediation, bioremediation, or nanoremediation. Yes, it is bioremediation. Next question. What is the genetically engineered microorganism used successfully in biotechnology to clean up oil spills? Bacillus, Pseudomonas, Nitrobacter, Nitrosomonas. Yes, Pseudomonas, which are used to clean up oil spills. A longer question. Why is DNA fingerprinting or DNA profiling better to be used than normal fingerprinting? Can identify the parents of an individual, can identify the criminal in a criminal case, can identify inherited diseases, cancer, and disease caused by virus, cannot identify babies born in the hospital that are exchanged with another person's baby. Yes, it is A. Can identify parents of individual and also can identify the criminal in a criminal case. Now, how can the DNA of an individual with a genetic disease be changed? Take a minute or two to think about it, and we can check your answers in a while. Yes, by gene therapy method, damaged genes are removed and replaced with normal genes. That's it. Okay, moving on to Hot's question. Now, in a murder case, a knife with blood stain was found by the police at a crime scene. By using DNA extracted from the sample on the knife, DNA profiles of the victim and 3 suspects were obtained as shown in figure 1. Okay, so now DNA in the blood knife, the DNA from the blood on the knife is very important. So whatever blood or DNA that is found on the knife should match the victim and also one of the suspect here. So the question is, among these 3 suspects, 1, 2, and 3, who is most likely the murderer? So what we can do is looking at the DNA on the knife, so we match with the victim and also the DNA from the suspect. Okay, so we're looking at this. We have suspect 2 over here. This bandwidth is from the victim and also suspect number 2. This bandwidth over here is also similar to suspect number 2. We have 4 matching bandwidth already. And then this bandwidth over here was also matching with this DNA from the victim and also suspect number 2. And lastly, the last bandwidth over here is also matching with the DNA of the suspect here. So, most likely to be is suspect number 2. The arrangement of DNA fragment bands of the suspect number 2 matches the DNA arrangement obtained from the blood sample on knife. Now, one of the identical twins is found to have committed a crime. Can both of them be distinguished by this DNA profiling technique? Yes or no? Why? No, both their DNA sets consist of the same nucleotide sequences. However, both of them can be identified by the differences in their fingerprint patterns. And so I hope you have benefited from this lesson and hopefully it helps you to do your revision. Thank you.